Thank you for joining us today for Spotted Lanternfly in Massachusetts, our August 2023 quarterly update. We're really pleased to have with us today Tracy Leskey, who's a research entomologist for the USDA. Tracy is actually a graduate of UMass Amherst, so she's from Massachusetts, but not here anymore, and we're really happy to hear the topic of our discussion today. But before we do that, we are actually going to switch things up a little bit from the way we usually do it and have an update from our SLF survey lead, Astra Perkins, who's going to give you an update about exactly what's happening with spotted lanternfly in Massachusetts right now. Once Astra's done, we're going to hear from Tracy about the threat of spotted lanternfly to specialty crops, the monitoring tools they're working with, and the potential for SLF to spread via human-assisted transport. After that, Elizabeth Barnes, our forest pest outreach coordinator, is going to talk to you guys about what to look for right now in terms of what spotted lanternfly is doing in Massachusetts, like what life stage it's in and things like that. And once we're done, we are going to have a question and answer period. We should be on to the SLF survey team update. So I'm going to stop sharing. Astra, if you are ready to give your presentation, we are ready for you at any time.
Awesome. Can you see that?
Not yet, but I can see it starting. There we go. Yes.
Perfect. So hi, everyone. My name is Astra. I am the SLF survey lead for MDAR, and I'm just going to give a really brief update about what we've been doing since our last webinar, which was in May, I believe. So to recap, and maybe it's new for some people here, currently Massachusetts has 4 known populations. The first one we found was in Fitchburg in late 2020, I believe. And then we found one in Shrewsbury in early 2021. And then we found one in Springfield, and then in Worcester. And pretty much all of these we found because someone reported seeing something through our website, and we went out to check it out. And lo and behold, we did in fact have a population in those areas. So here's a little recap of what they kind of look like. All of these red marks were positive points. A point is a host tree or host shrub, and then the 30 feet around it. Each one of these squares is a kilometer squared. So you can kind of see Shrewsbury's pretty contained and small, and then Springfield is a little bit bigger and has kind of these offshoots. And then Worcester has a larger area where we've also seen some outliers. And as we've been surveying this year, we've seen spread a little bit to the south in Springfield here. And then not so much spread in Worcester or Shrewsbury. And I don't have Fitchburg on here. It's actually the smallest infestation, and it really only encompasses 3 properties that are right next to each other. So it's just not nearly as big as these other ones. So early this spring, we installed bug barrier traps. Here's an example right here. It's just an inverted sticky trap basically, and the foam barrier pushes it out from the tree bark a little bit so that it works with the natural movement of nymphs, especially, which hatch in the spring and molt a few times during the summer. They tend to climb up and down the tree from where they were hatched out of their egg masses, and then they'll crawl down to more succulent material in the understory, and then they'll crawl back up the tree as they get larger and can get their sucker through the bark of the trees. So Springfield and Worcester have 30 plus traps right now throughout the infestations. They're usually in like the heaviest areas. We were using them mostly to try to mitigate and catch as many nymphs as we could so that they couldn't reach adulthood. It's, you know, it is what it is. It helps. We'd have to have probably hundreds of these out to really make a huge dent, but it does help, especially when we put it on trees that we know are heavy. Had a lot of egg masses that we couldn't reach when we scraped over the winter, etc. And then Shrewsbury and Fitchburg have traps also in the heaviest areas of population. They have fewer traps because they're smaller, and they have more kind of concentrated pockets where there's heavier populations. And then, so this summer we've been visually surveying outside of the edges that we established for these infestations by the end of last fall. We stopped our survey in probably early November. It had been cold enough at that point for all of the adults that were left to die off. And then we switched over to egg mass survey and scraping. So we've been out there on the edges of those infestations, starting from where we left off and moving outward to see if any of the insects had migrated. We're also switching those bug barrier traps over to these circle traps. These are just a little more effective at catching adults. They move up through this wire mesh and then they get stuck in the bag. And then we actually they basically just fry in the bags. We put them on the south side of the tree so that the sun gets them when they get up into these bags. And we've also been investigating public reports from the website. Nothing so far has led to any new infestations, thankfully. We still have a ways to go before the season's over, and we'll probably get plenty of more reports. But so far, no new infestations. We have confirmed that adults are out, especially in Springfield and Worcester. The Springfield infestation is like the most southerly and also kind of really urban. So they probably have a little bit more growing degree days. So they come out first that we've seen the last couple years. And then Worcester, we just confirmed adults as of Monday. And Shrewsbury and Fitchburg, we still haven't seen any, but there's probably a few around in Shrewsbury and Fitchburg. We're expecting them any day now. So we'll be out there surveying for those. So the Springfield population appears to have spread slightly as of the last time that we went out there in the fall. The adults do migrate quite a bit, especially after they breed, which is around now to mid to end September. They'll be breeding and then they'll lay eggs starting mid-September through to October, November when they die off. And they do kind of congregate at some points late in the summer to breed, and then they fly off to go lay their eggs wherever they want to. So that's why we see kind of spread in Springfield. In Worcester, it appears that we're not seeing quite as many as we expected, and we did do a lot of egg mass scraping this winter. So I'm going to chalk it up to we did a pretty good job with that, but probably what's happening is they've just migrated somewhere that we just haven't gotten to yet. But it does appear that we have fewer there, and the populations do ebb and flow through the years, just has a lot of variety of factors that go into that. And we do have treatments scheduled in Worcester, and we're working with property owners to schedule others in all 4 of the infestations. So that's basically what we're up to, and that's about it from me. So I will stop sharing. Maybe, did that work?
Yeah, thanks. Astra, before I know you have to go, I'm going to make an exception to saving all the Q&A for the end for Phil Lewis, who asked what specifically you're treating with.
So right now we're treating with bifenthrin. We only have a few places scheduled, and we want to just knock down the populations as much as possible, like right now. So we're using bifenthrin, but we do use dinotefuran as well at various times of the year, as Phil knows. But currently it's just the bifenthrin.
Thank you. All right, I know you need to get going, Astra. Thank you. If questions come up that are geared towards Astra that the rest of us can't answer, then we'll make sure to get an answer from Astra and email them out to folks. Thank you so much. I hope you have a good day. Tracy, if you are ready, I welcome you to share your screen and get started.
So good morning, everybody. My name is Tracy Leskey. As your hosts have said, I'm a research entomologist with USDA ARS, the Agricultural Research Service in Kearneysville, West Virginia at the Appalachian Fruit Research Station. So I'm going to talk about a couple different topics today. They include their threat to specialty crops, some of the monitoring tools that we've been working on, as well as spread via human-assisted transport. But I thought I'd start with, if I can get it to work here, there we go. Just a little bit about SLF. If you aren't as familiar with, this is an invasive species that is native to China, India, and Vietnam. And it's become invasive in the United States, South Korea, and Japan. And so at this point, and I'll talk about it later, it's been found in a number of eastern states throughout the US, and it continues to spread, unfortunately. This is just the life history of the bug. As Astra was talking about, we're getting to the point in the season where we have fourth instar nymphs. These are their oldest nymphal stage, molting to the adult stage. The adults then will continue to feed for some time. They will mate and they will begin laying eggs. And they can survive up until a heavy frost. Then these eggs will remain in the environment, and hatch begins. We've seen it as early as late April in our region, but it continues into June. And there are the four nymphal instars. The first three are the black with the white spots, and then they molt to the fourth instar, the red with the white and the black spots. So just a couple of pictures if you haven't seen them in the environment. So these are the younger nymphs, the first through thirds, the fourths, and then the adults, both with wings held over their bodies and also open. And this is just the egg mass. And certainly they are very nondescript, those egg masses. They look like a bit of mud sometimes on a tree. If you're looking for egg masses, a newly laid egg mass is going to look something like this with this waxy protective coating placed over it. Whereas old eggs from the previous year, that coating kind of flakes off over time and the eggs hatch. And this is what it's going to look like if you see them, already hatched and probably there for at least a year. Again, the first through thirds, these have black bodies and white spots. They're strong jumpers. That's one thing that always surprises me about all life stages as far as the nymphs and adults, how strong they are in terms of jumping. They have a very broad host range for the early nymphal instars, and we'll talk more about that, but they generally feed on the younger stems, smaller branches, and foliage to imbibe sap that they need to survive. When we hit the fourth instar, again, you can see that they have bright red bodies with black stripes and white spots, a bit bigger, again, strong jumpers. And this is when we start to see their host range narrow a bit. They're not feeding on as wide a number of host plants as they do in the earlier instars. This is just the adult stage again. Females tend to be slightly larger than males, and you can see one at rest here with their wings held roof-like. Over time, in the early season, they're fairly not as robust as they become toward the latter part of the season when they will begin laying eggs. So you can see that they really fatten up over time and they feed pretty intensely during the adult stage. One interesting thing is that we've even seen differences in the seasonal phenology just from Pennsylvania versus Virginia, where we did our initial work. We now have it in West Virginia too, but you can see the difference in just, several hundred miles on how the seasonal phenology can vary. So that's kind of just to give you some ideas of what SLF look like. What I want to start talking about now is the fact that this really is an invasive landscape level pest. And our concern has been not only can they be feeding and reproducing on native as well as invasive woody plants like tree of heaven outside specialty crop production areas like vineyards, but also, unfortunately, they can also feed and reproduce in vulnerable crops such as grape vineyards. And so when this bug hit, one of the things we were most concerned about were the host plants that it could attack. And there have been a number of studies that have documented host plant records over the course of the past few years. And certainly there have been over 103 plant taxa from 33 families identified, a number of forest tree species, and also the fact that while tree of heaven is often referred to as a highly preferred host, they can complete development in the absence of tree of heaven. But studies so far has shown that their fitness and survivorship were reduced. And a number of studies, including Duchesne, showed that over time, the number of wild hosts they're using narrows again as they mature to the adult stage. But again, tree of heaven, often referred to as a highly preferred host and an invasive plant itself. And if you look at the literature from Asia and Japan and Korea, where it's considered invasive, grapes, Vitis species, prunus, so peaches, as well as apples were considered to be potential hosts and also crops at risk. Wine grapes definitely have been the ones so far that we've seen impacts on based on some of the studies that have been conducted by our colleagues at Penn State in Pennsylvania. And so all of this information, though, led us to ask the question, or questions, I should say, what specialty crop as well as common wild host plants bordering specialty crop production support development and survivorship of spotted lanternfly? And how could the landscape mosaic that is really common in our region in the mid-Atlantic, and for that matter, Massachusetts, contribute to spotted lanternfly pressure in vulnerable specialty crop hosts, things like tree fruit and wine grapes? So when we first started working on this insect, it hadn't spread quite as far. And so we had some pretty strict quarantines in place, and we still do. But this work initially had to be conducted in a quarantine facility at USDA ARS at Fort Detrick, Maryland. And so this study was to look at what hosts can support survivorship over a 2-week period. And so we looked at early instar nymphs, later instar nymphs, and adults, and we provided them with a single host. And this is the list of the hosts. And some of this work was funded by the Forest Service to understand what forestry species also could be at risk. So we had tree of heaven, black cherry, black walnut, and black locust. We had common hackberry, sugar maple, white oak, mulberry, and we had 2 specialty crop hosts, peach and apple. And these were conducted over 2 seasons. So in these studies, the early instar nymphs, we did see a significant difference in survivorship. But greatest survivorship, not surprisingly, was on tree of heaven, followed by black walnut, peach, and sugar maple. But on all other hosts, even in that time period, survivorship dipped below 50%. So we were already seeing that some of these hosts probably weren't very good. In the later instar stages, again, tree of heaven and black walnut supported greatest survivorship, whereas everything else, all of those other 4 species, dipped below 50%. And then you can really see the impact of host narrowing in the adult stage. There was good survivorship, again, on tree of heaven, but everything else fell below 2.5% by the conclusion of the trial. So really, we didn't see good survivorship on any of these over a 2-week period when they were only offered a single host. The next thing we wanted to do was look at the same kind of question, but with what we were calling the viney host. So we wanted to look at grape, in this case, a muscadine grape, Vitis rotundifolia, the variety is Carlos, hops, as well as kiwi. And so again, we started with this same design where we looked at early instar, late instar nymphs, as well as adults. They were provided the single host for 2 weeks. And again, these were all conducted in quarantine. And what we saw was for the early instars, survivorship on kiwi was not very good. It fell below 50% at 7 days. And it's probably because for the early instars, the kiwi vines are very hairy and it's really difficult for them to feed through. When we got to the later instars, there was, again, significantly greater survivorship on Tree of Heaven compared with other hosts. And then by the time we got to the adult stage, all hosts fell below 50% before the end of the trial, with the exception of Tree of Heaven. So again, most of these hosts weren't supporting their survivorship very well for a 2-week period. So that's just survivorship, though. We really wanted to get at the question, can they complete development on some of these hosts? And so we were looking at both specialty crop hosts. In this case, we had apple, peach, and that grape species, the muscadine grape, as well as black walnut and Tree of Heaven. And then we also had mixed diets of Tree of Heaven plus black walnut, apple, peach, or that muscadine grape. And in this case, we would start with 30 newly hatched first instar nymphs, put them in these cages and track their development for 2 years. What we found was over time, not surprisingly, again, Tree of Heaven had the highest survivorship, but in fact, all of the Tree of Heaven diets, mixed or single host, had strong survivorship. We also saw good development and survivorship on black walnut. And so if you look at the percentage that survived and made it to the adult stage, what you can see is Tree of Heaven just about 50%. These mixed diets had between anywhere from 25 to 50%, whereas peach actually only got them to the second instar at 6%. Nobody made it beyond the second instar. Apple, about 3% made it to the third instar, and then everybody died. And the muscadine grape, only 6% made it to the fourth instar, and everybody survived. In the case of these mixed diets, what we did see was they developed faster than the Tree of Heaven alone diet, which was kind of interesting. But one thing that really came out, and many other studies have shown a similar thing, is while black walnut can support their survivorship and development to the adult stage, once they are adults, they can't survive very long on just black walnut alone. And you can see it's significantly lower survivorship in the adult stage compared with all those Tree of Heaven diets, whether a single diet or a mixed diet. So some interesting observations from this study about hosts and host quality. And this kind of led to a follow-up study where we wanted to know what would happen if we did the same kind of survivorship and development study, but rather than having Tree of Heaven kind of be that anchor host, what if we used wine grapes, because wine grapes seem to be being attacked at that point, Vitis vinifera, versus the muscadine that we had already used alone or in combination with apple, peach, black walnut, in this case, silver maple, as well as Tree of Heaven, and these black walnut based diets as well. We wanted to see if they could complete development and survivorship when these hosts were paired together to try to understand the threat to wine grapes. And so what we found was, in the case of these wine grapes, we were seeing much better survivorship than we had with muscadine grapes. And these mixed diets seem to do pretty well as well, with the exception of peach. Peach is just not a good host. And then with black walnut, while some were completing development, it really wasn't, the survivorship wasn't as strong. And so if you look at the percentage that reached the adult stage on all of these different diets, you can see wine grape alone, Vitis vinifera will support their development to the adult stage. Along with these combination diets, we had individuals reach the adult stage, except for grape plus peach, where they only made it to the fourth instar. Versus the black walnut combination diets, some did make it to the adult stage, but the only one that really supported strong survivorship was when it was paired with Tree of Heaven. So Vitis vinifera or wine grapes appear to support development more strongly than rotundifolia, the muscadine grape. And so that was really interesting to us. So we wanted to look then a little more deeply at the vulnerability of different Vitis species. And so we went back and did the same kind of trial, but in this case, we looked at grape-based diets of Vitis vinifera, both a red and a white variety, a wild grape, riverbank grape, Riparia, a juice grape, Labrusca, and our Carlos or muscadine grape and Tree of Heaven as a positive control. And so in this study, what you see is Tree of Heaven provided the greatest survivorship, but it was equaled by Riparia, the wild grape, which tells us this is another host that this insect can use in the environment. Pinot Noir also supported strong survivorship along with Chardonnay, where you use Labrusca and muscadine, not so much. And you can really see that in the survivorship to the adult stage. We had Tree of Heaven, the vinifera, the Riparia, and Chardonnay, and then these really fell off the map. So certainly there are hosts that are better for spotted lanternfly. And what we found from this sort of suite of studies that most SLF nymphs and adults can survive on single specialty, asking the question, can they survive on these single hosts over a 2-week period? And most could support SLF for a short period of time, especially during the early instar stages. But by the time they reach adult, their host range really narrows, and there really are only a few hosts that can support their survivorship as a single host, like Tree of Heaven. Now, in terms of development on single and mixed host diets, we see good development and survivorship on Tree of Heaven, wine grapes, Vitis vinifera, riverbank grapes, Vitis riparia, and black walnut. Whereas rotundifolia and Labrusca were not good hosts, and nor are peach and apple good hosts. So we're much less concerned about those attacking tree fruit, which was our concern when they first invaded our region. But what it means is that they can complete hosts on wild and cultivated hosts. They can feed on a wide variety of plants during the early instar stage. We know from other research that Tree of Heaven, as well as our own, is not required to complete development. And so this mosaic of landscapes will likely enable them, unfortunately, to establish in new areas. And certainly wine grapes are a vulnerable specialty crop in terms of attack from this host. And we've seen that locally in our wine grape vineyards in Northern Virginia. But what that means is that we still then need to think about how we can monitor them as they potentially move into crops, but also spread across the landscape. And so I thought I would take you through a little bit of the work that we've done to study this insect and develop monitoring tools for this pest. And you'll see some of the same pictures that Astra showed, and it's based on some of the research that we've done, as well as our colleagues at APHIS, to identify traps that work really well for this pest. And so initially, a lot of these standard sticky bands, which work to capture SLF based on what we call their negative gravitactic behavior, which essentially means they like to move up, they work well, but they also capture a lot of non-targets. So we started experimenting with these circle traps that we've used in tree fruit pest management for a weevil pest called plum curculio. And this is the design that was original to that pest. Then we developed a sort of a modified trap with this polyethylene screen and a commercial prototype, which was made of a wire screen. And we did these trials both in Pennsylvania and Virginia with our colleagues at Penn State, counted adults and nymphs weekly, and we also measured non-target captures. And so, there we go. So what we found was that the modified circle trap and the sticky band generally perform best in most scenarios. These are from Virginia, each of 2 years, and Pennsylvania, each of 2 years. But the sticky band captured about twice as many non-targets, something we really want to avoid. These are the non-target captures. You can see 895 in the sticky bands. So things like, unfortunately, birds and butterflies and pollinators and other critters. And so we kind of moved then to these circle style traps to work with in terms of monitoring the presence, relative abundance, and spread of SLF in the environment. We also tried to understand if this commercially available lure methyl salicylate would improve trap captures. This was a simple system where we put posts in the ground to take away the potential for any host plant competition and evaluate this lure. The commercially available methyl salicylate lure was then deployed above these sticky traps, and we had baited versus unbaited controls. And these were checked weekly, and their positions re-randomized. We caught a lot of stuff, but unfortunately in the environment, we just don't see a strong response to these lures. And so there's still a lot of effort from USDA ARS and USDA APHIS to really identify some better olfactory stimuli that can be used with these traps to enhance their sensitivity and increase our capacity to do monitoring and biosurveillance. So that work continues. We also did a study over a couple of years to just figure out how can we optimize trap deployment, because it's not just about putting the trap out there, but at what height do you put these on trees? How often do you need to sample? Are there particular host trees that are better than others? And how do they work under sort of low relative density versus high relative density situations? And again, we worked with our colleagues in Pennsylvania, and we put these out in Virginia, and we did this work over 2 years. And what we found was, despite the fact that adults often sit at the base of trees, a 1 meter height for adults did not make versus 0.5 meters did not make a difference for them. But for nymphs, definitely 1 meter was better, at least numerically. In terms of trap sample interval, while you can see that there, again, wasn't any significant differences among them, we do see that numerically it's better at 1 week. And the other thing that happens, if you're doing this for monitoring, if you're just trying to trap them out, it doesn't matter. But if you're doing this for monitoring, the samples, the individuals will degrade over time and it gets a bit smelly. So we typically try to monitor on a weekly basis. In terms of host trees, we saw that for nymphs and adults, definitely Tree of Heaven and Black Walnut were best, but other hosts also routinely captured spotted lanternfly in 2021. And so if these two hosts aren't nearby, these data provide the opportunity to use other hosts such as hackberry locust or box elder to monitor these pests. In low and high population densities, we saw that these traps all worked reasonably well. Again, our circle traps were just as effective as sticky bands, and they didn't lead to as many non-target captures. And then the replaceable bag that you see, we got to evaluate that in 2021. We saw some pretty big differences under low and high population. Definitely under high population, we were getting a lot of nymphs in that replaceable bag. They can just accumulate in there. So that was good. So I think from the standpoint of, especially for trap out, those are really good traps to just sort of collect and collect and collect. So in terms of our key results that we've found from this study, we found that trap height did not significantly affect captures, but 1 meter was numerically better. Trap sampling interval, it didn't significantly affect the captures, but there is a yuck factor if you're actually trying to monitor numbers and count bugs. Host tree deployment, definitely Tree of Heaven followed by Black Walnut were best, but there are other hosts that could be used if they're not available. And then finally, relative density, the bag trap did lead to degraded specimens, and the sticky traps yielded more non-target captures. But again, it depends on the purpose. Are you trying to monitor or trap out? And if you have a periodical cicadas emerging, those also can be a problem when you're doing this kind of work. So that's what we've learned to date for trapping. But the problem is that not only are these bugs spreading through their natural tendency to move, both adults flying and adults and nymphs jumping, they also can be transported by humans. And so we know that these bugs sometimes are kind of indiscriminate on where they'll lay eggs. You can see them. This is a lamppost. This is a metal stake in the ground under someone's deck, cement blocks, all sorts of places. And so to deal with this, both extension folks in areas where this insect is quarantined have created these residential checklists so people can look for where these egg masses might be laid so they can be destroyed before they move objects if they're moving, you know, outside a quarantine zone. So these are really helpful. But what it doesn't take into account is the potential for nymphal or adult movement. And so if you look at the current known distribution of spotted lanternfly, one thing that has become sort of apparent is that they are following major corridors, major highways and railways as they spread. This is Interstate 81 that goes right through our region, and you can see the spread on either side of this highway. This area across Pennsylvania has a major railway as well as the Pennsylvania Turnpike. And so we're pretty certain that human-assisted transport is happening, not just with eggs potentially, but also with the mobile life stages. And so here's an example of one of our first seasons studying these insects with, this is a female who's about ready to lay some eggs sitting on a railway, waiting for a trip to go somewhere. So this sort of got us thinking about human-assisted transport. And if you look at human-assisted dispersal of invasive insects, there have been a long historical record and a lot of work done to try to reduce this potential for movement in cargo, obviously plant material, trains, ships, and vehicles. For example, the emerald ash borer and the don't move firewood and the Asian longhorn beetle not to move firewood. So there's a lot of examples of this kind of thing. And so we wanted to understand if lanternfly could accidentally be moved. So asking the question, could accidental hitchhiking by SLF on vehicles be an important dispersal mechanism and a pathway for spread? And so we developed this human-assisted dispersal bioassay. When we looked in the scientific literature, there really wasn't a good example of something that we could follow to do this kind of work. So we collaborated with our engineering department to use this laminar flow fan, a hydraulic motor, and we could place the insects on the hood of a vehicle and essentially look at what speeds they were being dislodged to try to get a sense of how long they might be able to cling to a moving vehicle. And specifically, when we created this system, can we measure adhesion and dislodgement by hitchhikers? And so this is an anemometer that we would place at the different locations to look at exact wind speed. And there were 5 areas that we tested in this original trial: the side panel, the windshield, the wipers, the well as the hood of the vehicle. And we looked at kind of 2 different behavioral states. We had acclimated spotted lanternfly, where they were placed gently on the hood with no wind flow, and non-acclimated, where they were just dropped and the wind was already moving 3 to 5 kilometers per hour. And so in terms of the acclimated, all of the mobile life stages were tested, the 1st, 2nd, and 3rd and 4th instars, as well as early adults and late adults. And once they were they were placed on the hood, we started cranking up the wind speed, and it took about 30 seconds to reach max speed. And then these non-acclimated, once they were dropped and the wind was already moving, we immediately just ramped up the speed to maximum. And so in terms of the results from this, what we found was that acclimated versus non-acclimated, fairly similar patterns. We didn't see a lot of difference, although there's more variation. These are violin graphs. So it gives you an idea of where most of the individuals landed in terms of the speed at which they were dislodged, as well as the variation around that value. And so with the non-acclimated, there was definitely more variability. Also, interestingly, we did see that females were able to cling a bit longer than males in this study. In terms of just overall life stage, and this was for the acclimated individuals, what we saw was that first instars and early adults were able to cling the best at the highest speeds, versus the later adults had the poorest adhesion in terms of this study. And then if you look at the percent of each life stage reaching the maximum wind speed, what was interesting is that in every location, some individuals, maybe not of every life stage, could reach the max wind speed. Even on the hood, 7.3%, and this was first and early adults mainly, reached the maximum wind speed, which is about 60 miles per hour. The side panel, we had a number of different life stages, the wipers, the windshield, and the well, but definitely the two places where these insects could cling the best were the wipers and the wiper wells, where we had just about half able to reach maximum wind speed, and that means they could be transported. If you look at kind of their posture, there is a difference. And if the insect is kind of sitting up, as you see here, this seems to result in dislodgement, versus successful adhesion is when they're kind of already hunkered down, a little bit more flattened. They seem to be a little bit more aerodynamic. And other factors that may influence these results are just their life stage. So what we see in the earlier life stages is there's not as much of what they call this terminal sticky lip, which is this little adhesion on the base of their feet, which in the adult, it seems to be more suction cup-like. And then the width of their feet, the aurolium size and shape, the adults have just a wider base in general, it seems, whereas the fourth instars, you can see it's very narrow. So we think that these morphological differences may influence the results. So certainly from the standpoint of, from the standpoint of movement, first instars cling the longest along with early adults. First instars, it was the windshield, but for early adults, it was the wiper and the wiper well in particular, where they could remain the longest. This was just us looking at the sort of the eddy and flow of the wind. But all life stages were able to reach maximum wind speed at some locations on the vehicle. And so this makes it more likely that human-assisted transport is likely. So we're working with collaborating modelers at Temple University to create a model that takes into account this transportation-assisted SLF spread, so that we can try to define areas where we should be looking for this pest. And I think the other thing that's really important to think about with spotted lanternfly and with invasion ecology in general is that it's the rare events that are important, because all materials had some SLF that did not dislodge. And so what that means is that, you know, some of these individuals could be taken for a ride. We have people, that video that I showed right at the start of my talk was sent in by someone to show that they were driving down the highway with a spotted lanternfly. So something we need to keep in mind, and if possible, and this is what we've always done, we check our vehicles and blow them off with a wind blower, a leaf blower, when we would be working in quarantine zones. Oh, I have to give a warning now. Okay, so last slide, I'm just going to wrap up, but I just want to say, so the other piece of this story is what happens when a lanternfly is dislodged from a vehicle. If it hits a windshield or something like that, does it survive? So I'm giving you fair warning that the next slide shows what happens when lanternfly hit a windshield. When we looked at survivorship of this for the modelers, these were data. So I just want to show you this, but again, I don't want you to, this is work that we've been doing in the lab to get at this transportation model. So you can see that some individuals unfortunately meet their demise, but in other cases, they kind of just spin off and they survive. And so we see about 50% of these individuals surviving. So with that, I'm going to wrap up and just say thank you for your attention. Our funding sources, my colleagues with the Stop SLF project, and my colleagues at USDA, ARS, and USDA APHIS. And with that, I'm going to stop sharing and I'll turn it back to Jen and Elizabeth.
Thank you so much. Just a reminder for folks, if you have questions for Tracy, I see a few have come in already. Please put them in the Q&A. I am going to turn things over to Elizabeth for the final segment of our webinar.
I'm just going to wrap things up real quickly now. So just as a quick reminder, this is the point in the spotted lanternfly life stage where we are right now. The adults and the fourth instars are the main things you're likely to be seeing. Those fourth instars, as a reminder, are bright red with black and white markings on them. So they've got little dots all over their back. They do look a little bit like a ladybug, but they are bigger and they have white spots instead of black. The adults are going to be a gray brown color with spotted wings. When they're at rest, those beautiful bright colors are mostly hidden except for those two little dots right below their eyes. However, when they are in flight or dead or dying, or sometimes if you disturb them, they may flare their wings and show off those bright colors that you're probably used to seeing in pictures and make them look almost like butterflies, even though they're not. So those color patterns are, there's a stripe of bright red with some black spots, a swoosh of white, and then black at the tip. And as was mentioned earlier, when they are getting ready to lay eggs, the females' abdomens will swell up and turn this beautiful bright yellow color. Okay, and I wanted to mention the egg masses as well because pretty soon they're going to start laying eggs, certainly before our next webinar. The eggs are a gray-brown color. They contain each usually around 30 to 50 eggs, and they have a putty-like coating over most of them. Sometimes you find them without that putty-like coating though, so you may see these seed-like little eggs. They generally start laying eggs here in mid-September, although it can be a little earlier or a little later, kind of depending on the year. All right, now, what do you do if you see spotted lanternfly? Send in a report. And the first thing you should do when you spot that lanternfly is get a picture. Before you move, before you go anywhere, pull out your phone, pull out your camera, whatever you've got with you, take a picture, doesn't matter how close you are to it. Then sneak a little closer, get another picture. And after you've gotten those photos, you know, if you want to smash it, if you're positive it's a lanternfly, go ahead and do it at that point, but get that picture first because they are pretty good at hopping away when disturbed. And that picture is incredibly important to us in terms of confirming there's a lanternfly. You know, send in a report either way, but if you have a picture, that's even better. Also note your location. And those are, you know, the two big pieces of information that are most helpful to us. Now, if you aren't sure if it's a lanternfly or something else, still send in a report. The people who review the reports, I'm one of them. We love bugs. So if you send in a photo of something and it turns out not to be lanternfly, you know, oh, well, maybe you've sent us what turns out to be a cool photo of a native moth. We are perfectly happy to see it. As long as it's, you know, a good faith report, we will just let you know what it is, maybe include a cool bug fact in there. Now, I always want to emphasize too, if you see something weird, and by that, I mean, if you're someone who spends a lot of time outside and you find this bug you've never seen before and you can't identify, or if you notice all of a sudden a bunch of a specific type of plant are dying in a way you've never noticed before, ask someone about it. That can be us. That can be your friend who's just really into insects. That can be, you know, just posting it on iNaturalist, but just make sure you ask someone about it because that's how we catch a lot of new invasive species that we weren't even aware were in the country. So you can find that linked report at the bottom of the page and the QR code. That works for all of the invasive species we're talking about. We'll also post all of the links in the chat in a moment. And I did also want to mention we have some resources. If you are in Massachusetts and you are in a position to do some sort of outreach, we are happy to send you free spotted lanternfly materials. Those include mini posters, flyers, scraper cards, stickers, all kinds of fun stuff. You can also download those off of our website. And if you want to do your own presentation or design something on your own, let us know about it. We're always happy to provide photos as long as you give us credit. I also wanted to mention we do have an invasive pest dashboard that shows the areas in Massachusetts that have In this case, the orange shows where spotted lanternfly have been found in small populations, and the blue show areas where we found individual lanternfly. And you can also find similar maps for kudzu and beech leaf disease and all sorts of other interesting things on there. So the link is at the bottom of the screen again. And I also wanted to mention we are doing a survey to figure out how we can improve the dashboard. So if you have just a couple minutes, we would really appreciate it if you would fill that out. There's a pop-up screen when you first, or not a pop-up, but there's a little window that shows up when you first go to the dashboard, and that has a link to the survey. It's also in the chat here, and I will send it to Jen as well so that she can post it in the chat on the side. And we would just really appreciate it if you fill that out, even if it's your first time looking at the dashboard. We have a newsletter, goes out once a month, 5 bullet points or less, timely forest pest information. We try and keep it short and sweet with links out to further reading if you're interested in learning more. You can sign up here. Webinars are archived on our YouTube channel. Again, the link will go in the chat for that. If you want to share today's webinar with someone, that'll be up shortly.
And we're now going to move on to the question and answer session of the webinar. One of them that came up that I thought it would be good to address right away is someone was asking, would you consider using chicken wire over the sticky bands to help reduce non-target collections? And before I let Tracy answer, I do want to make it clear that we do use sticky bands. You saw the picture that Astra showed you, but ours have a covering over them. When we use, sorry, she was showing you those. Never mind. We always use a covering over ours to reduce non-target collections in Massachusetts. Tracy, do you want to speak to the work that you've been doing?
Yeah, I meant to say that when I noticed that you guys had one of the trap designs that reduced those non-target captures. So yeah, I think I've seen other people do the chicken wire thing, and I think that helps a lot with some of the, especially the bigger vertebrates, but you still might get more, some of the smaller pollinators and things like that getting through your chicken wire. So it's just something to think about. I think we're all trying to do our best with this kind of thing, reduce those non-target captures. So chicken wire definitely will keep the bigger critters away.
We also do advise the public to use to avoid those non-target captures as well. So we know that, like, we're really not at the stage yet in Massachusetts where the volume of lanternfly is so strong that people are going out of their way to set traps like they have with things like spongy moth in high infestation years. But in the outreach that we've prepared, we do make it clear, like, you need to cover this with mesh or chicken wire or something. So, all right. Somebody asked another question that I thought was a good one. Did the frost that we had in February, we had a really deep freeze in February, did it kill off the egg masses? I mean, I can say that we know it didn't because we know it didn't kill all of them because we had significant hatch, and the eggs are always hatching the season after they're laid. But I didn't know if, Elizabeth, do you want to add anything, or Tracy, either one of you, feel free to contribute.
I mean, you covered it pretty well, I think. That they're pretty hardy over the winter, at least in terms of the level of cold that we get here in Massachusetts. So they were fine with that frost. We've still seen a decent number of them this year.
Yeah, we haven't, unfortunately, at least in our region, had temperatures over an extended period of time that we think has led to a lot of mortality to date, at least in, since I've been working on them. So.
Thank you. Someone asked, how do you destroy an egg mass besides scraping it? Elizabeth, I mean, smashing and never mind, you take this one.
Well, I mean, so scraping it really is the best way to do it. And do I have a card handy? Okay, so I don't have a card handy. So let me show these sticky notes and pretend these are a scraper card, but you want to just like crush them first and then gently scrape them off. There are also some horticultural oils that you can use as well. And we do on our website, we have a management guide that talks about that a little more, because there are a few modifications in how you might use them just in terms of timing and things like that, compared to other things you might use it for. So I would check for that. I'm trying to think, I mean, I think those are the two big ways I know of to destroy egg masses. I don't know if you know of any other options, Tracy.
No, like really, we're down to stylet oil beyond scraping. So that's been one of the better, the mineral oil is a better one. We did have chlorpyrifos, but that's not permitted anymore in food use situations. So that one was good against it, but it's not labeled for that so any longer. So yeah.
Thank you. So someone asked, in the aerial maps, the SLF reports seem to overlap with developed areas more than forested or green space areas. Is that because there are more traps there, or that there are more people who can potentially report? Elizabeth?
That's okay. So it's an interesting question. And it's always tricky to kind of tease apart because, on one hand, we would expect to get more reports and be more likely to find spotted lanternfly in areas where there are more people just because they're more likely to notice them there. But we do have traps set up all over the place, not just necessarily in places where there are a lot of people around. On the other hand, though, they're more likely to be transported to areas where there are a lot of people going in and out. So where there are big transportation corridors, where there are people commuting a lot, where there are people traveling from infested states, or there are trains or, trucks that are, shipping packages and equipment and all of those things between states. Those are more likely to get spotted lanternfly as well. So it can be kind of hard to separate those two things sometimes. But yeah, I mean, that's where we've seen them. And, in Massachusetts, yeah, that's kind of where we've seen them popping up. The other thing is we don't tend to see them in like deep forested areas that much, like in the center of a major forest. They tend to stick closer to the edge for the most part. I mean, that's what we've seen. Correct me if it's different where you are, Tracy.
No, I think that's where they get started, usually because tree of heaven grows very well in disturbed soils. Unfortunately, as they've continued to, the populations have built, we have seen them deeper into forests in some areas. So it's kind of a little bit of both. I would add that as public awareness becomes more apparent too, the likelihood of being sighted in new areas also goes up. So, there's that factor too. Yeah.
I was just responding in writing to somebody asking about where to report, because I realize there might be people wanting to report from other states. Keep in mind, there are some states where SLF is so widespread, like in New Jersey, that they're not really fielding reports from folks anymore. But in Massachusetts, as of right now, we still are. There are a couple of folks asking about like egg mass treatments. Does bifenthrin work to kill eggs or egg masses, or could you use neem oil? And I know that they've been, like, a lot of folks are working with something called golden pest spray oil, which is made from soybean as a treatment for egg masses. I feel like neem could possibly be effective, but I don't know off the top of my head if that's being widely used right now. Anyone else, please jump in while I clear out these questions. I don't think bifenthrin would work on egg masses, but I'm not sure. So I guess we need to figure out the answer to that.
I will say, as a plug for our archive, we had someone in, I think, February, I want to say, who did talk about egg mass treatments and what they've been finding with egg mass treatments. So if that's something you're particularly interested in, I'd recommend Again, like I mentioned before, we have that documented on our website that goes over various treatment methods. Penn State also has a lot of incredibly good information on their website. I strongly recommend checking out them because they're, kind of on the front lines of this. And then also, again, that webinar in our archive is a pretty good overview of treating the egg masses.
Yeah, I mean, most people are recommending, one of those oils because it's a little more environmentally friendly and it does smother the egg masses pretty well. So, yeah.
Yeah, I mean, Elizabeth, if you could throw a link to the Pennsylvania guide in there, it might into the chat.
I can't share things in the chat.
What?
Yes.
I am very sorry about that. I do not
worries.
know why that is happening. You're supposed to be able to. All right, I'm gonna get that while you guys answer another question. Somebody asked, after removing host trees, what is the best way to remove an infestation? That is a loaded question, I think. Who wants to take it?
So
No, Tracy, great.
I can talk about what we've thought about. Unfortunately, with Tree of Heaven, it's not an easy one to remove because even after you cut it down, it sprouts and it's terrible. So we've been trying to do some work with colleagues at Virginia Tech and WVU who found a verticillium wilt that kills Tree of Heaven. And if that gets to the point where it's labeled, because it's very specific, it kills Tree of Heaven, it hasn't shown any, effect on other tree species, you could take out their hosts more easily. But the issue is that even with Tree of Heaven gone, some of the work at Penn State has shown that they can complete development on other hosts. We know that grapevines, particularly Vitis vinifera, they can complete their development and they do well. So because it feeds on so many different hosts, it's really is not, I think, maybe taking out Tree of Heaven could help, but you still have all these factors that we don't quite understand. And there could be other hosts that we haven't even identified yet that could support their development and survivorship. So we're looking more toward biocontrol as an opportunity for both Tree of Heaven as well as the pest itself. So I think that it's something that can operate across the entire landscape and not just, in a particular area.
Thank you. I found the management guide link for folks that really want to do a deep dive into treatment options. And I found the switch that makes it so panelists can also share links in the chat. Apologize for that. Oh, someone just asked, what was the oil the doctor recommended for the egg mass treatment? And maybe that we just answered that, right? The golden pest spray oil.
Stylet oil.
Oh,
Horticultural
stylet
oils.
oil. Oh,
well, there's a couple different formulations that have been evaluated, as I recall. But the Penn State guide should give you exactly what they're recommending. So I'd take a look at that.
Thank you. All right. And you're saying stylet oil is in the mouth parts of the insect, right? Okay.
Yeah.
Just checking. Someone was asking, is there a threat of tree removal similar to the management of Asian longhorn beetle? Elizabeth, do you want to take that?
No. So because of what we just talked about, there's not, we don't have plans at the moment to do tree removal. It doesn't seem to be effective. And if we were to do it, it would be with Tree of Heaven, which is invasive anyway. But like I said, that's not in the list of things we're doing at the moment. There were a few trees that we removed in Fitchburg when we were first working on this to see if that would be effective. But I don't think we've done that at the other sites. Correct me if I'm wrong, Jen.
We did some limited tree removals in Shrewsbury. It was at the very early stages of the infestation. It didn't do what we were hoping it would do, which is remove the SLF from the area. It's not an effective way of eradicating SLF, and it's expensive, and Tree of Heaven is, I don't know if persistent is really the word to
Invasive.
describe. Yeah, it's just, I mean, it fights back if you try to remove it.
One thing, oh, I just want to add just on this, one thing I just remembered that some of the folks at APHIS had done in early days was when they took down trees that had a lot of egg masses, they put them through a bark or wood chipper, and that was very good at destroying egg masses. So that's, you know, if you have a tree of heaven on your property covered in egg masses, you want to run it through a tree, apparently a wood chipper, it'll take those egg masses out and you know, pulverize your tree of heaven too for good measure. So
Yeah, but yeah, they're very, spotted lanternfly and Asian longhorn beetle are, you know, they're very different species. They have very different life cycles. So Asian longhorn beetle, because they're mostly contained within those host plants, that's a very effective way of getting rid of them. Whereas spotted lanternfly, you know, they're moving all around. They have just so many host plants, and it's not just trees. I mean, we find them pretty frequently on poison ivy.
yes.
So they will try to eat almost, I don't want to say just about anything, but like an amazing variety of different host plants. So that's not something that, tree removal isn't something people should be concerned about with spotted lanternfly.
I do want to note that it's 11:18. We say we go till 11:15, but I would like to continue, and I believe the panelists have agreed to stay on a little later to answer a few more questions. If you have to go, I totally understand, folks in the audience, but Tim had a question. Let's see. He was asking, so I think Tim must be, all right, he's in New Jersey. I was going to say he's not in Massachusetts. He was saying, you know, why do they find them on their cell tower? Is it the warmth? And then mentions that he was using Gorilla Tape to catch them, I guess, on the cell tower. And I see Tracy nodding. So do you want to take this one?
So there are a couple of things. They seem to orient, especially adults, they seem to orient to high points. We have a monument here in town where it sits above the Potomac River, and it's just it's like a beacon. It's like an antenna for lanternflies. So they seem to orient to tall objects. We know that from some of the visual ecology, like you'll find them on, you know, telephone poles and things like that as well. And in fact, Penn State was doing some work to exploit that behavior. And there also may be a humming sound. There's been some work by Miriam Cooper Band that they may respond to these vibrational sounds and signals. So maybe the tower's providing the right frequency. So a couple different things could be going on, but we know for sure that they go for tall, upright silhouettes, dark silhouettes against a bright background. I had my undergrads collecting lanternflies for an experiment we were doing this past week, and with drones, and they went to the monument and, you know, got a couple hundred in 5 minutes. They're all there. So, yeah.
Some folks were kind of asking about natural predators. I know we get that question in a lot of these webinars. Anybody
want to
take
go, Elizabeth? I can wait.
Sure. Yeah, so it's so many of our native predators will eat spotted lanternfly, but I think that question is getting at, you know, do they eat enough that it's actually going to control it? So yes, they will eat it. You may see birds eating them, spiders, ants, all sorts of different, you know, insects and vertebrates will eat spotted lanternfly, but they don't eat enough to control them. So in order to control them, you would probably need something like a parasitoid, which is a specific type of insect that in this case, we would want to find one that would only target spotted lanternfly and nothing else, which is something that actually they're working on right now to try and identify a parasitoid that's just going to go after spotted lanternfly. And if we could find something like that, we could maybe, you know, we're not going to completely eradicate them, but we could maybe get their population under control in such a way that they're no longer such a huge problem. But that's something that's way down the road, needs to go through a lot of testing first to make sure they're just going to go after spotted lanternfly and not any of our native insects first. So something to kind of hope for the future, but not rely on at the moment.
Thank you. So I'm trying to multitask, which is kind of failing, but someone asked about similar looking, things similar to the egg masses. So I wanted to put in the chat our egg mass lookalike guide that MDAR recently put out. And then I figured we would also want to share the one for adults and nymphs. So, you know, Massachusetts has put together lookalike guides specifically for folks in Massachusetts, like species they're likely to see, like tiger bee flies and leopard moths and things like that, that we know people are going to report. Someone asked if the summer rain has affected the life cycle of spotted lanternfly. I mean, it hasn't stopped it. Things were, we had like a bit of a slow start, I think, but it hasn't impacted their lives. You know what I mean? Like they're maybe a week behind where they were last year, but
So one thing that you might see that we saw in 2018, where we had rain like you guys are getting this year, we had a big entomopathogenic fungi outbreak, where we had a couple different species. They were, you know, it was so damp and yucky, they became infected. So maybe you'll get lucky and get some of those ento fungi taking out a few.
haven't seen much yet, but still hoping.
Yeah.
Here's a good question for Tracy. What are the effects of SLF to cultivated high bush blueberry crops? Have there been any observations or damage reports?
Not that I am aware of, where we've seen them feeding on blueberry. I think that, you know, it just so far, nothing to report. We've looked at other crops, and there've been, but mainly it's if they have kind of a viney structure, like we've seen kiwi, they'll feed on hops, grapevines, but a lot of the, and things like some of the vegetables, you know, like cucumber and things. But as far as blueberries, nothing that I'm aware. And even some of those other crops, even though they will feed on them, doesn't seem to be very good hosts, at least to date, you know.
Thank you. All right, let's take a couple more questions. I want to finish up here. Someone did ask how you tell the difference between male and female SLF.
Yeah.
Go ahead.
The tip of the abdomen on the females, it's a little red valvifers. They're like little red piece that sticks out on the tip of the female. Males don't have it.
Okay. Would you say also that like body shape changes on the females?
Oh, they're giant. Like by September, they're like trying to hoist themselves up in the tree or not going very far.
This is a really interesting question. Is there potential for the evolution of SLF as selection through survivability? Like eventually they might be better at surviving on plants that aren't tree of heaven?
I mean, you know, you can never count out biology, but I don't think so in the short term. There seems to be something very specific about tree of heaven that provides them with increased survivorship and fecundity and longevity. But I don't know, because there are other things out there that, like I said, they may not have to evolve if they find the right host locally that supports their development. So yeah,
That's a really good point.
Yeah.
I pulled up some data while you were talking about all the different, it was really, really interesting to see all the different grape species that you tested. And when I looked, I mean, most of the grape we see around here is Labrusca. So I was like, okay, that's good. But then we have a lot of Riparia here. Like I would say it's probably the second most common wild grape species. So, oh, well.
Yeah, it just, I mean, it's more about them being able to establish in some unmanaged areas, you know, with the Riparia being present. And that was why we wanted to test it, because it's common here as well.
All right, so we have two more questions here and then we're going to finish up. Well, I think this was probably answered already. I was just skipping around a lot. Treatment options available for homeowners and our licensed applicators. I'm going to say we already answered that. I'm going to throw another link in the chat to the management guide that we did so that you guys can see that. And then the final question was, do they kill plants and trees or just use them to spread? Who wants to take that?
I'll tell you what we know so far, and I'll turn it back to Elizabeth. We know from some of the work, and more of this is going on, we haven't seen them, even when we force feed them on things like peach and apple, it doesn't kill them. But we have seen, for example, when we force fed them on peach, they had reduced capacity to deal with frost. There was increased frost injury, but again, it's not a great host, so I don't expect any issues. But on wine grapes, some of the work from Penn State, they saw a decrease in there was an increase in winter injury, and it's probably to do with their capacity. The adults feeding a lot in the late season, they weaken the plant to some degree, making it more susceptible to abiotic stresses. You know, we've seen like some, like Tree of Heaven, we'll see it kind of droopy and yucky and stuff, but if they leave, then sometimes the plants return. So we haven't necessarily seen anything die yet, but we have seen some declines, I would say.
Thank you. And then Elizabeth, I'm going to let you finish up, but I just wanted to warn you, I'm going to share the final slide while you're talking. So you're just so you're not caught off guard. Go ahead.
Gonna disappear in a minute. Yeah, I mean, I would agree with that entirely. It seems to be there are some plants that they have a stronger effect on than others, and those tend to be the plants where they really, really like them, like the Tree of Heaven or the grape. It also, you know, depends on how healthy the plant is going into it, because they are causing this stress on the plant. So, you know, if it's really healthy, if it doesn't have a lot of lanternfly feeding on it, it's probably going to be okay. But if it's already stressed, or if it has heavy lanternfly feeding and then there's another stress, that can potentially start to cause some issues for the plant. So yeah.
Thank you. All right. I think we're about at the end here. Thanks for everybody who hung on till the end through what I thought was a really interesting Q&A. There's a lot of links in the chat. We'll try to send those out to participants too, just so that, you know, folks that want to learn more about specific treatment options or sign up for outreach materials, get that information. Just one final reminder, if you're looking for continuing education credits, we will process those and get them out to you by September 16th. So just give us a month to get through all that data. There were a lot of you here today. Our next quarterly update is going to be in November. If you would like to register now, I put a link in the chat. There's also a link up on the screen here. And I just realized that I did not give you guys the exact date. So hold on one second while I attempt to get that information. It is November 15th at 10 a.m. So with that, we're going to wrap things up unless we have anything else from our speakers. Thank you, Elizabeth, and thank you, Tracy, for joining us today. I think all of us learned a lot, including the panelists. Thank you to Astra, even though she's not here anymore because she's out in the field. And we hope to see you guys in November. Take care, everybody.
Bye.